Hello people. So platelets are actually platelets or thrombocytes are not regarded as cells but cell fragments. These cell fragments are being produced in the bone marrow from their parent cell, the megakaryocyte. The megakaryocyte is a big cell. It has actually a diameter of 50 to 150 micrometers, and it has this lobulated nucleus. The megakaryocyte can release platelets explosively, so fast, or continuously. And actually, the proposed mechanism in explosive release is actually that the megakaryocyte fast releases a lot of thrombocytes into the bloodstream. And in this continuous release, the megakaryocyte has this function of producing these platelet ribbons. And from these platelet ribbons, platelets are one by one being released into the bloodstream. So they are going from the membrane of the megakaryocyte into the blood vessels, and then one by one these platelets are being released. A megakaryocyte can have about 3 or 2 of these platelet ribbons in their lifespan, and one of these ribbons can release about 2,000 thrombocytes into the bloodstream. So a megakaryocyte can release about 4,000 to 8,000 of thrombocytes in their lifespan into the bloodstream. The thrombocytes are cell fragments that are small in diameter, about 2 to 4 micrometers, and in a cubic millimeter of blood we have about 150,000 to 400,000 thrombocytes. The thrombocytes are actually shaped like a disc, and if we make a section through the middle of the thrombocyte, we will see something like this. So the thrombocyte has this peripheral zone, this zone is rich in glycoproteins. These glycoproteins are important for the thrombocyte activation, adhesion, and aggregation. Then we have this zone. This zone is sometimes regarded as a marginal band. And this zone has microtubules. These microtubules consist of a protein that is called tubulin, and it maintains this discoid shape of the thrombocyte. It also has microfilaments. These microfilaments are actin and myosin, and these filaments in an unactivated thrombocyte, they are monomers, and when the thrombocyte gets activated, they are polymerizing and contracting. This zone is the periphery, and if we go further in, we will see that we have this organelle zone. In this zone, we have remnants of organelles that are actually from the parent cell, the megakaryocyte. We have the endoplasmatic reticulum, also mitochondria, then all these granules that are important for the thrombocyte function. We have the biggest and the most numerous are the alpha granules. So these are the biggest and most numerous granules in the thrombocyte. Then we have delta granules, and we have small lambda granules. These granules have in them molecules that are important for the function of the thrombocyte, and that is blood clotting. If we now take what is in these alpha granules have, for example, fibrinogen. It is also factor 1. Fibrinogen is important because if prothrombin, a protein in the blood, gets activated into thrombin, thrombin then cleaves fibrinogen into fibrin. And fibrin is an important part in the blood clotting machine. Then these granules also have the von Willebrand factor, this factor is important in adhesion of the thrombocytes. Then also they have thrombospondin, thrombospondin is a protein that has anti-angiogenic functions, so it doesn't allow new blood vessels to form. Alpha granules also have the platelet derived growth factor. This platelet derived growth factor is an important stimulant in proliferation of fibroblasts. So all these molecules are important, and proteins are important in facilitating the function of the thrombocyte. That is actually the stopping of blood going out from the blood vessels into the tissues or into the outside. The delta granules have in them serotonin, molecules like ADP and ATP, and these molecules are important in facilitating thrombocyte aggregation. Serotonin is also, it has many functions, but it has also a function of vasoconstriction. Lambda granules have enzymes, and they are regarded as thrombocyte lysosomes. So, also, these endoplasmatic reticulums that the thrombocytes have from the megakaryocytes have in them calcium ions, and they also produce thromboxane 2. All these molecules together are important and function somehow in the formation of a clot and stopping the blood going out from the blood vessels into the tissue, which is the primary function of thrombocytes. So if there is a damaged endothelial wall, the thrombocyte will be activated through the glycoproteins in its periphery zone. There will be a polymerization of actin and myosin filaments. There will be a contraction and a degranulation of these granules. All these molecules will then activate the thrombocyte. The thrombocyte will then aggregate and form this primary clot, which is its primary function. Thank you.